Assalamualaikum warahmatullahi wabarakatuh. Good evening, everyone. Today, we would like to present our presentation about how pathogens affect plants, part 2. But before going any further, let me introduce our group members. First is me, Faizalyah Putri Jasmine. Second, Syifa Nurfadlika. Third, Safina Mutia Husna. Fourth, Meira Safitri. Fifth, Lilin Sefianingrum. Sixth, Ananda Sulistyatama. And the last is Nanda Agustian Rohmadi. So the first material will be read by me. Although pathogens enter plant cells mechanically, the activity of pathogens in tissues is mostly chemical. Chemical compounds produced by pathogens consist of enzymes, toxins, growth regulators, and polysaccharides. These compounds are important pathogenicity factor for causing different diseases between pathogens. Types of substances produced by pathogens. Enzymes. Enzymes are biomolecules that function as catalysts or compounds that speed up the reaction process without being sped up in a chemical reaction. Enzymes are biocatalysts, which means they can speed up biological reaction without changing their chemical structure. GPT. Growth regulators play an important role in controlling biological process in plant tissues. Its roles include regulating the growth rate of each network and integrating the parts to produce what we know as plants. Toxins. Toxins is defined as any chemical substance that causes a harmful effect when administered to an organism. This is distinguished from the poison that is produced in the body of an organism or living thing as a result of metabolism, which is called a toxin. Polysaccharides. Polysaccharides are carbohydrates that have long polymers and are composed of hundreds to thousands of monosaccharides. Polysaccharides are naturally found in plants and boromic acid in the cell walls of bacteria.
Role of pathogen produced substance in pathogenicity. The first is enzymes. The reservoir of pectin produces a fluid collection of pectin substance that unite plant cell along with a weakening of the cell wall, encouraging tissue aberration, or plant tissue becomes soft and loose, and cells separate and eventually die. ZPT. ZPT in Indonesian is abbreviation of growth regulator. Important growth regulators include tamoxifen, which naturally resides in plant in the form of IAA, gibberellin, cytokinin, and ethylene. IAA is necessary for elongated growth and differentiation. Gibberellin can accelerate growth, stimulate flowering, root and stem lengthening, and fruit growth. Toxin. Pathogen that produce toxin to kill host tissue such as Sclerotinia mushroom, Botrytis fusarium, and Pseudomonas bacteria. Sclerotinia sclerotium infects stem, leaves, and head tissue through the development of hyphae from germinate ascospores. The flowers are a place of food supply to support the infection carried out by sclerotiums. Polysaccharide, not too much of role than others. Enzyme. The wax coating on plants affects the degree of pathogen colonization. Some pathogens, such as Fuchsinia hordeae, produce enzymes that can degrade wax, while other pathogenic fungi and penetrate the wax layer with only mechanical force. Cutin is the main component of the cuticle. Many fungi and some bacteria produce case on and/or esterase that can degrade cocaine. The production of cutinases can be increased by some of the fatty acid present in the wax layer that binds to the cuticle on the cuticle. Nevertheless, the presence of glucose suppresses the expression of the quantity gene therapy, lowering the production rate of cutinase. Cutinase can be inhibited by specific inhibitor compounds or antibodies on plant surface on plant surface. For example, phenolic compounds such as chlorogenic and caffeic acid that can inhibit the quinine monilinia fructicola cause chocolate rot in stone fruit. Antilipase antibodies can inhibit the penetration of Botrytis cinerea fungi, which produce two cutin degrading enzymes, namely cutinase and lipase.
Crude regulators. Crude regulators are organic compounds that are not nutrients. GPT in small amounts can spur, inhibit, and can change the psychological process of plants. Growing regulatory substances make an important contribution in the world of agriculture. Plant hormones are a collection of organic compounds. Both are both natural and man-made. And now type of growing regulatory substance is first, auxin. Auxin is a plant hormone that serves as a regulator of cell enlargement and trigger cell lengthening in the area behind the meristem. Auxin can be used to spur plant growth. The hormone auxin is naturally found in the roots and of stem and flowers. Cytokinin. Cytokinin serves as a trigger for cell division in plants. Compounds that can function as cytokinin are kinetin and zeatin. Kinetin was originally found in vultures' sperm extract. Natural zeatin can be obtained in young corn kernels. Zeatin is also found in coconut water. Gibberellin. Giving gibberellin under the title of a plant can increase the rate of synthesis photos. Plant leaf plant leaf develop significantly because this hormone spurs leaf growth. There is an increase in cell division and cell growth that leads to leaf development. In addition, it also encourages the lengthening of plant stems.
Ethylene serves to aid the process of fruit ripening, encourage flowering, stimulate flower expansion, stimulate root and stem growth, stimulate fruit and leaf abortion, stimulate seed germination, inhibit the lengthening of sprout stems, strengthen plant stems and endormancy. Next. Reacon tunnel. Until now, the working The working mechanism of this hormone is not 100% known. In various studies, triacontanol can increase the ratio of acidic sugar in citrus plants and can increase tea production. The result revealed the administration of triacontanol with low concentration in corn, tomato, and rice crops can increase growth. Next, inhibitors. Inhibitors play a role in inhibiting root growth. The application of hormone inhibitors is utilized. There is a type that help the enlargement of the bulbs. For example, in potato plants, carrots, onions, and so on. Its application is done when it will prevent the growth of new shoots to enlarge plants' bulbs. Paclobutrazole. The use of paclobutrazole is intended so that the tree can bear fruit outside of the season. The hormone paclobutrazole inhibits the biosynthesis of gibberellin. Vegetative growth of plant is stunted and spurs generative growth. When vegetative growth is stunted or stopped, flowers will appear and bear fruits.
And then toxins. Toxin is a compound produced by microorganisms and toxic to the plants. The toxin reach on the protoplasm of a living host. Toxin generally damage cells by impairing the permeability of cell membranes, interfering with enzyme work, or acting as anti-metabolic, thereby causing a deficiency of essential growth factors. Agrios 2005. Toxins can be classified into two groups as follows. Number 1, non-specific host toxin. This toxin can damage many types of plants and even from a variety of familia. Example, top toxin, tentoxin, bacillotoxin. Top toxin. This toxin produced by Pseudomonas ringe tabaci that cause the rust disease in black on tobacco. Top toxin not toxic, but once inside the cells hydrolysis, top toxin be toxic. Tentoxin. This toxin is produced by fungus Alternaria alternata or longipes, which causes rust spotting on tobacco and symptoms of orange patches on dragon fruit. Bacillotoxin. The toxin plays a role in pathogen virulence by breaking down the host plant disease resistance caused by hollow black bacteria, but also fungi, bacteria, and other pathogenic viruses.
And then number 2, host-specific toxin. This toxin can affect only a few plants of varieties that become the pathogen-forming plant of the toxin death. There are several kinds of host-specific toxin. First, the toxin, HV toxin, HC toxin, and EM toxin. T-toxin. The toxin is produced by the fungus Decelera maedis rasti. This toxin causes leaf blight in corn. The toxin specifically damages mitochondria. HV toxin. HV toxin is formed by the fungus Sociobulus helminthosporium victoriae. This toxin causes the disease leaf blight and eventually destroys the entire plant. This toxin also causes change in the structure of the cell wall and others.
HC toxin. HC toxin is produced by Concleus or Helminthosporium arboreum. The toxin cause of leaf spotting disease on corn. This is only toxic to specific corn derivatives where sensitive corn is deficient in this corn, so it cannot protect itself against this toxin. EM toxin. EM toxin is produced by the fungus Alternaria alternata pathotype of apple. This toxin causes Alternaria leaf spotting disease in apple. This toxin highly selective against sensitive apple varieties. Polysaccharide. The main role of polysaccharide appears in weathering disease with pathogen in fighting plant vascular tissues. Polysaccharide release pathogen on xylem may cause blockage causing withering. However, it's believed that polysaccharide synergize with macromolecules compound released by pathogen so that plant become weathered. Thank you. Any question?